Good morning. I'm worried that you might think that it's impossible to give a talk on this topic, but that's why I have to give it, because most of what you hear in public, and even in the scientific public, turns out to be complete bullpucky. People think in a very lazy way about origins, and my message to you today is that it's like any other scientific question. It can be settled by experiment, and I hope to persuade you of that in the next few minutes. But first, a word from our sponsor, Giordano Bruno, who was, as Glauco has said, he was fried in the Campo de Fiori.
Roasted. No.
He was roasted.
Yes.
He was roasted in the Campo de Fiori with a wooden lock on his tongue, because he said things that were unacceptable. He looked up in the sky. He was, in fact, one of the first astrobiologists. He looked up in the sky. Among the unacceptable things he thought was, when he looked in the sky, he recognized the lights there as other suns like ours, and thought there would be planets around those suns with beings on them. An amazing thought for the 15th century. So hello again, Giordano, and in some ways we celebrate, in some ways the course celebrates his insight. So to try and formalize what I just said, the origin of life is a scientific problem. It can be settled scientifically. We have the means, the intellectual means, to investigate it and to settle it, to solve the problem. And that's what this is about. Okay, so you read a lot of things. You can go anywhere. You can go to any magazine, and every magazine, no matter what its subject is formally, ultimately will have an article about the origin of life. And if you read those articles, you'll find that life begins when you first have heredity, when you first have metabolism, when you first have membranes, when you first have Darwinism, or maybe when you first have fur. But my message to you, and my assumption, is that all these people are right. And you must understand that my assumption, because of my background and my taste, is that the origin, when you say, what is the origin of life, you're talking about finding a biomolecule that reproduces or can be reproduced, and it confers a phenotype. If you can get to that point, I quit, I retire. I have solved the problem of the origin of life. And these guys are right too. If you want to say that you've got to have metabolism before you have the origin of life, that's fine with me. That's also correct. We have to understand each of these events to make a coherent narrative that leads you to life as we see it today. And so everybody's got the same logical status. But when you hear me, you're hearing about this, okay? And you should understand that I also understand that that's arbitrary. Okay, so first of all, where did the molecules that compose life come from? Well, to an amazing extent, I think, we can identify these as things that just fall out of the sky. They're cosmochemicals made in the cosmos. The most amazing of which, and the only one I'll talk about, is the discovery of Juan Aró, who was a Catalonian, and if he were still working, would attend this course maybe, that you can take 5 HCNs and make adenine. The 4 HCNs go together to make this diaminomalleonitrile. That closes to make the 5-membered ring that you have in purines, and then whatever, you can then close the big ring, the 6-membered ring of a purine, depending on what small molecule you have around, And you readily make, in rather high yield, some exotic purines like xanthine, but the standard purines, most notably adenine in the highest amount, but also guanine. So you make the biological purines with with 5, perhaps, of the most prevalent small molecules in the entire universe. If you look out through you take the spectrum of the solar nebula, HCN is there. So unmistakably, this has meant to people for a long time, since 1962, when Wanneroo published this. This has meant to people that life somehow utilized the stuff that was around in the forming solar system, and that we still believe today. Now I'm going to talk about ribonucleotides. I'm influenced by this adenine story, but there's an even more powerful argument from continuity, that we ought to be talking about RNA. And that I can describe you as an inspection of the timeline of the Earth. So here's the Earth today. Here's the formation of the Earth, whose dating we know pretty accurately to the 3rd significant figure because of the dating of stuff in asteroids in the solar system and other old materials. And the amazing thing is, it is really an amazing thing. I can still remember being stunned by this when I found it. There is complex life in the universe, complex life on Earth, the only life we know about in the universe, very shortly after the planet congealed from the solar nebula. So here, at 3.8 billion years before the present, or I have it in giga years ago, GYAs, a little bit of an exotic unit, but if you drop that into a conversation, you will provoke more conversation. Anyway, the 3.8 giga years ago, we have organisms that are fractionating the isotopes of carbon like modern ones. So you have metabolism and a complex metabolism and effective organisms right here. So the RNA world, which we think of as just prior to that, must be back here somewhere. Maybe these arrows ought to be fuzzy rather than sharp. Somewhere back in here must be the origin of life. In other words, we have complex life. The simplest life has got to be somewhere in here between the time the Earth forms, and actually we know it's in this direction because back here is the moon-forming impact, which liquefied a large part of the crust of the planet. So life can't be happening until somewhere in here. So we know the origin is there a long time ago, and we can date it approximately. But that's what intimidates people. People say, 4 billion years ago, I can't even go into my lab and study things that happened yesterday. How are we going to do this? And as I said, my message is, we know how to do it. Don't listen to people who say that we don't know how to do it. So usually when we study a problem, we do it for the last 400 years anyway. We've known how to find out which things are true. And let me say to you as members of this course that that's another speech, which I won't give, but I want to point out that you are the custodians of the true. The idea that something can be true is being degraded all over the world, but scientists know what it means to be true. It's true from here, it's true from there, it's true no matter what you do, and that is not a widely shared perception, but it's an important one, and it's in your hands, and we've known what it means for at least 400 years, because 400 years ago, Sir Francis Bacon said, men have sought to make a world from their own conception. And I'll leave you to read it, but what he says is, just bring it into the lab and look at it. Find out what it actually does. And that's the road to true knowledge. So experiment, since Bacon we've known, is the way. But the trouble is, a lot of interesting things, indeed I would say the most interesting things, you can't do experiments on. You can't do experiments on where the universe came from. You can't really do experiments on ecology. If you're interested in bunnies in the forest, you can't make a forest and fill it with bunnies. You can't study evolution. In fact, anything with a historical dimension is outside the Baconian definition of what science does. Because if it happened, you know, if we were with, we can't be with Giordano Bruno in 1600, but even more so, we can't be around to see what happened 4 billion years ago. So some of these things, the origin of life, origin of the code, evolution considered broadly, have a historical dimension that takes their study completely out of the normal framework. But I've been saying, maybe you've noticed I've been saying, that it doesn't take it out of a scientific framework because there's a way to study it. So let me tell you a little bit about the Reverend Thomas Bayes. Thomas Bayes, he published one paper in his life. But was a fantastically important paper. So that's encouraging to those of us who are scientists. Maybe if we just get the right paper, and we publish a lot of papers, and hope that one of them is like Bayes. So Bayes gave his paper to a friend, and what was in Bayes' paper? Well, so this is a little. So now I'm going to get a little abstract, but I'm not going to place big emphasis on it. What I want you to do is understand what this means. So this is a construction that every person who has taken a statistics course has seen. This is called a sample space, and the points in here are events. And the bigger the area you have, the bigger the probability is of your event, right? So you can, so event B is probably twice the probability of event A. Event B, if event B is having lunch, maybe this is getting coffee at the break, or something like that. Anyway, so the thing I want you to appreciate is that both circles share this red area, this intersection we can call AB, and just using that simple perception alone, I can go in one step to what's called Bayes' theorem. There's no doubt about Bayes' theorem. If you can get somebody to bet on Bayes' theorem, you have a sure win, because it is the most basic thing in the theory, in the calculus of statistics, or the calculus of probabilities. And here's what it says. It says that rigorously, you can have some opinion. You can have some opinion, I should say. This is the opinion you start with. And you can update that opinion to a new probability by just asking what the probability of the evidence that you see before your eyes is, given your opinion, over the total probability of observing that evidence. Well, that's a lot of words, but in some ways, this is an absolutely intuitive perception. Bayes' theorem, we call it. So you look around at what you observe, and it isn't some kind of weak-minded conclusion to say that the most probable history, including the most probable history of a planet, is that history that leads to what you're seeing. That's Bayes' theorem. Common sense, right? Nobody in the room is going to bet against that. Nobody is going, no policeman, no prosecutor is going to bet against the idea that the evidence that he has most likely indicates some one course of events leading to the present. Everybody will agree on that. But what Bayes showed is that that's obligatory, that it's quantitative, that you can calculate probabilities. I'll come back to this. This is my next point. This is the important one for us as evolutionists. And obstacles become your friends, because if there's something you can't explain, if there's something you can't explain, I'm getting pretty excited here. There's something you can't explain, it means your hypothesis is incomplete. So here's the important thing, and this looks like a formula. In fact, I think I have to concede that it's a formula. But here's the important point. Here's Bayes' theorem again. Never mind. These little vertical bars mean given that. So here's the probability of my hypothesis. Given that, I observed data 1. I love it. I mean, I really do love it. And what you can do is you can substitute in to show that if you have data, you have one set of data, you have another set of data, you elevate the probability of what your hypothesis is that you started with, but you elevate it multiplicatively. So if you do one experiment, the probability goes up. You do 2 experiments, the probability goes up. And the probability that you originally started with goes up by the product, okay? So that's the thing that is not present in common intuition, and that's why you can study something as abstruse as the origin of life. If you elevate the probability of one origin by tenfold and then tenfold, you have elevated the probability that you started with the correct hypothesis by 100-fold. So because of that formal fact, we can study events that we can never do Baconian experiments on. And what that means, what can study means, is that we can take some hypothesis and make it much more probable, which is all we're doing with any experiment. So that's why I started out by saying that we can study the origin like any scientific problem. We can make a hypothesis about the origin much more probable than any other idea that we have. Scientific success, what can I say? Okay. But it's better than that. It's easier than that in some peculiar sense of the word easy. It's easier than that because I'm going to argue to you that when we have the solution, we can recognize it. We'll know when we're done. And we start from a perception of Darwin's. So this was in the first edition of The Origin of Species, 1859. Darwin says, well, probably, you should read it, probably life started from one origin. There was one creature. And that all, therefore, every modern organism is descended from that creature. We now know that that is exactly true. Darwin retracted that, incidentally. It was too provocative to the church. It disappeared from later editions. But we know that it is completely true more surely than we know almost any other fact in science, not just in biology, but in science as a whole. And the reason we know that is because of the big tree of life on Earth. So Carl Woese and George Fox, 1977, they had, due to an inspiration of Carl Woese's, they had a way to compare the sequences of creatures' genes. It's a little bit tricky, and there's still a lot of discussion about this. You've got to take a gene that's not exchanged between organisms because you want the sequence to reflect the history of the organism. So genes coming in from another organism are obviously going to be confusing. And this is not trivial, but their insight was you could just compare sequences and count differences and make lines that were the length of the number of sequence changes, and you could make a tree. And the Woese-Fox tree was the first to show something that, once again, there's still discussion, but I think is unquestionably true, that all life on Earth falls into 3 related groups, and you know what the relation is between them. There are archaea, there are bacteria, and then over here there are creatures with nuclei, which most people in the course are most interested in. So 3 lobes and a big tree on which you can put everything that's ever been sequenced on Earth. And now the impressive point is that they only had a few hundred sequences, Woese and Fox, and now we have millions of sequences. Millions. And maybe that's even out of date. Maybe it's tens of millions of sequences. You all know how big the sequence databases are. And so here's, this I think has hundreds and hundreds of sequences. It doesn't even begin to show the millions of sequences that we have, but it is still true. It is still true that there's a 3-lobed tree, 1, 2, 3 lobes, that are bacteria, they're archaea, or they're eukaryotes. It's still true, and every sequence, every one of those million sequences is recognizably related. In other words, as Darwin incredibly said, well, there was one origin for life on Earth. This is actually is ribosomal RNAs, so we're really talking about the translation system. So I should say there is one translation system on Earth, but that sounds really dorky. So I'm going to say there is one origin for life on Earth. The translation systems in everything, elephants, giant viruses. It all, when you have one that's big enough to have a translational sequence, they're all from the same origin. It's the only reasonable explanation. So what we're trying to do is we're trying to get back to this point. In fact, but we're trying to get back behind that point, before that point existed. That's the last common ancestor for all life on Earth, Luca. We'll discuss Luca again, but keep him in mind. I guess maybe it's her. Keep him or her in mind. We'll come back to that. But that's what we're trying to do, is get back behind that point. And in summary, 4 billion years, reducing environments, oxidizing environments, every environment on Earth, there is one successful origin for life on Earth. Okay? Suppose I asked you, what is the origin of the motor car? Okay? When you look out the street, there are electrics and hybrids and things that burn hydrocarbons, but they've got roughly the same number of wheels, and they work in roughly the same way. What does it mean that they all look alike? Well, in this case, we know what it means. Carl Benz, in 1885, made his patent Motorwagen, which he sold. Actually, his wife, Bertha, invented about half of the things on the car that were important. She invented the lined brakes that we still use. She invented the transmission. But Carl invented the carburetor, the spark plug. And every car that we now have has this same sequence of innovations. And so it is absolutely clear that every car on Earth is a descendant of Carl Benz's car in 1885. And you can go look at it. You can see, even without inquiring into all these mechanical details, that it's true. We have a much more powerful constraint on the origin of life on Earth. A carburetor is impressive, but it's not as impressive as thousands and thousands of nucleotides that are almost the same, which is the case for all the organisms on Earth. So the conclusion that we can trace Earth's biota back to an origin, unmistakable. And when you see, we can find the origin of cars, I think for exactly the same reason, we can define the origin of life. Okay, because this idea that the origin of life is an ordinary scientific problem is so different from what people often say, I'm going to do an example, an extended example that has some handles that everyone has thought about. Everyone's thought about the dinosaurs. So, but it was 65 million years ago. It's 10 times as long ago as our last common ancestor with chimpanzees. Nothing like a primate existed on Earth. There were the most primitive mammals at the time, but it's out of reach. It's almost out of conception in the same way that the origin of life is. So it's interesting to think about why we think, and we do think, we know something about the cessation of the dinosaurs. So it was a mystery, though, until the 70s, when the hypothesis by Louis and Walter Alvarez was that the dinosaurs had been killed off by a giant impactor was proposed. And part of the evidence is that in layers, and these are layers I'm familiar with in Trinidad, Colorado, near my home, in layers up to a certain very sharp division in the geology of the area, there are dinosaur fossils, and then they just stop. They stop dead. You never find them in rocks that are younger than these rocks where there's a so-called iridium spike. So iridium's a heavy metal. There's almost no iridium on the surface of the Earth because when the Earth's core formed, the iron core took down all the heavy metals. So heavy metals very low in concentration on the surface of the Earth. Iridium is high, though, in things that never made a core. So in asteroids or other debris in the universe, iridium that's flying around the universe, iridium's very high. So dinosaur fossils up to here, then an iridium spike, then no more big lizards. Birds, still dinosaurs today, but no more big lizards. So what does that mean? So the Alvarezes recognized what that probably means was that the iridium was brought in on a huge bolide that hit the Earth. And it was the consequences of that collision that killed off the dinosaurs, the reason there was never another dinosaur. Well, of course there's been fantastic amount of controversy, but there's a lot of evidence, and that's my point. So I briefly want to flash through some of the evidence just to show you how one of these arguments goes. Well, there are these things called tektites. They're glasses that are thrown out from a place where, any kind of place, but usually an impactor where a lot of energy is released. So these things are melted glass, and they're found radiating from Yucatan, where this thing hit. So they're evidence for the collision. These lines are what you see in quartz that's been hit with a very quick, low temperature but high pressure impact. And all the quartz around that area has been shocked. The most impressive evidence, the unlikely thing that you would find in the absence of this hypothesis about the big impactor, is that there is a crater, which I'll show you in just a moment. But for example, the Yucatan Peninsula, so this is the Yucatan, Mexico's down here. The Yucatan Peninsula is covered with sand from marine sources. So that's evidence for huge tidal waves, which of course accompany a 10-kilometer wide impactor, which is what we're talking about. And here is, as the final kind of thing I want to consider, the final kind of evidence for a huge impact at 65 million years ago, 66 million years ago. Here is the scariest data I think I have ever seen as a scientist. So NASA flies over. They have instruments that measure the gravitational field. They have instruments to measure little fluctuations in the magnetic field. And they can combine those into a map of the underlying strata. So here's the coast. You can't see it very well, but here's the coast of modern Yucatan, and underneath the coast is this 180-kilometer wide crater. And most of the pictures you see, in fact, the picture I showed you to begin this section of the talk, are wrong. You can see, I mean, it just makes the hair stand up on the back of my neck, I must say. You can see the impactor coming in at a very flat angle because it made this trench in Yucatan before solidly smacking into the crust of the Earth. And you can see a plume of ejecta coming out, like the tektites that we were talking about, like the iridium that ended up in Trinidad, Colorado. And you can imagine, in a way you cannot imagine in the presence of any other kind of evidence, you can imagine the huge event. Okay. So my purpose in going through this is to come back to this idea that you can multiply, you can multiplicatively elevate the probability of a hypothesis with independent kinds of data, just the way you always multiply probabilities for any kind of independent event, right? So here is Senor Rex looking out some morning, I think it's a morning. Let's see, he would be looking north. Well, I can't tell you where that orb comes from. Anyway, he's looking out, and here comes an impactor, probably at not a flat enough angle. But anyway, he's looking out, and we think we know what his fate was because of these different things that interlock like a jigsaw puzzle. There's only one way for them to go together. This can only be put together one way. So that's an intuitive way of expressing the fact that when you have multiple kinds of evidence, they have a multiplicative, not an additive effect, but a multiplicative effect on what you think the explanation of Senor Rex's long vacation really is. Okay. So let's call that a Bayesian convergence. We have a Bayesian convergence on the impact that killed the dinosaurs. Still discussion. Some people think it's volcanoes in India. Indian volcanoes. Indian volcanoes are good. They definitely existed at the same time. And the latest, I think, is that people believe that the geophysical impact of the cratering of the big impactor in Yucatan on the other side of the world actually increased the volcanism. So the volcanism might be it was definitely there before, it was there after, but it might have increased from the impact itself. Anyway, there's a very authoritative paper by 50 people who study these kind of things and the dating, who say, no, the extinction is coincident with the impact. Forget about all this other stuff that occurred before. which you can read in Science in 2010. So these things go together in a convincing way. Can we imagine that we might generate a Bayesian convergence then on the origin of life? We need it. We need it. You are students of RNA, and there is a crisis there is a full-on crisis in the theory that there was once an RNA world. Some of you are not feeling moved by this crisis. In fact, some of you are asleep. But there are others of us who worry about these things. For example, the idea that there were once creatures who used things like ribozymes. Boy, it's tough to convince yourself of that in the laboratory, because ribozymes are composed of things that have unique linkages. They're regiospecific, only have 3', 5' phosphates between the nucleotides. So how you would ever get those is a problem in a world that has no catalysts. The notable work has been done by Jack Szostak on that topic, and maybe regiospecific's not so important is the moral of his recent work, one moral of his recent work. And so I'm taking this list from a great review he wrote. Duplexes of RNA are very stable. Gee, when you try and replicate RNAs in the world of 4 billion years ago, you would have a heck of a time getting things that are the size of a ribozyme apart. So how are you going to get them apart? Oh, okay, I'll just use helicases. Well, how are you going to involve helicases? So the high TM, the stable duplex, is a problem. How do you, there's a problem with fidelity. How do you on a rock, on a forming Earth that's, just a few million years ago, had a collision with a Mars-sized body. How do you make accurate copies? Replication, if you get too slow with replication, it's not fast enough to keep up with degradation. So even if you have some RNA, it immediately goes away faster than you can reproduce it. Nucleotides are not particularly stable. Not good for the hypothesis. The substrates hydrolyze. Divalents, which you need, like magnesium, to give RNA-specific structure, speed up the degradation and wreck membranes, so it's hard to see how to make cells. Even if you could replicate, how you're not going to make the primers for replication internally, because that's making another complex hypothesis about what your protocells can do. So the fact that you can't get primers across membranes is important. If you manage to get through all these things and make a ribozyme-sized piece of RNA, which is a copy of a pre-existing piece of RNA, the product inhibits any further progress with the template that you started with. So product inhibition is a terrible problem. In my next lecture, I confidently predict that I will solve all these problems simultaneously with my hands in complete view the entire time. So I promise you to do that. But the solution is that but let me tell you what the solution is instead of standing up here and doing stand-up. The solution is that ribozymes are too big, that all this stuff about stability and accuracy and all that stuff goes away if you're trying to make something small. So what's wrong with the RNA world hypothesis is not that RNAs didn't do it. RNAs did it. I will bet you that RNAs did it. But they weren't the RNAs we're thinking of. Knowing about, the self-splicing RNAs and the processing RNAs like RNase P. The RNAs that we have to talk about were smaller. And that's what I'll do next time. And I will try to answer any questions at this point if you have them.
Thank you very much.